Bueno, bienvenidos, bienvenidas. Ya vamos a empezar. Okay, so it's a real pleasure for me to introduce you to Dr. Alexandre Gaspar Maia. He's an associate professor in the lab of medicine and pathology at Mayo Clinic, and he's also the director of Epigenomics Development Lab at the Center for Individual Medicine, also at Mayo Clinic. He's Portuguese. He earned his PhD in molecular biology from the University of Coimbra in Portugal. And he also performed his thesis project at the UCSF in stem cell biology in the lab of Miguel Ramalho Santos. He did his postdoc training in the lab of Emily Bernstein in Mount Sinai in New York, where we actually met. And he studied the role of histone variants in several aspects, and reprogramming also focused on cancer biology. In 2017, he started his own research lab at the Mayo Clinic, and he focused on functional epigenomics and works on various aspects of transcription and enhancer regulation with implications on cellular heterogeneity and drug resistance in cancer and cellular reprogramming. He also has numerous awards. In particular, he got 2 postdoctoral fellowships from the USA Department of Defense when he was working on Mount Sinai. And he also got an NIH Career Enhancement Award from the Mayo Clinic, and the Breast Cancer Sport Award in 2022, and the Young Investigator DoD Ovarian Cancer Academy Award. He's an expert, as you can see, in single cell genomics and how to apply these techniques to know how to cure cancer. So I'm pretty sure he's going to be an awesome talk, and well, thank you for being here.
Thank you. Is this good? Okay. Hi, everybody. Gracias por la invitación. My Portuñol is pretty good. So yeah, I met David in New York when I was a postdoc, and he was a grad student, and so it's really nice to see how he's going to start his new adventure here. So today I'm going to be talking. About a very recent work that we basically had just submitted a couple of weeks ago that delves into our use of single cell sequencing to understand cellular heterogeneity in the context of ovarian cancer, and specifically in chemotherapy resistance. But I know that I know that this is a aging institute. Obviously, the cancers that we're going to be talking about, ovarian cancer, is mostly prevalent in aging women. But I think the technology itself could be really interesting. We have a very big aging center at Mayo Clinic that we actually started collaborating very recently. So I hope that from the things that I tell you about, you will probably get inspired for some of your work as well. So the title of the talk is Deciphering Epigenomic Signature in Cancer Progression and Chemotherapy Resistance in Single Cell Resolution. But before I really start into the project itself, because I know that there's a lot of students here, I also kind of just wanted to give you an overview of my path. Also to remind you guys that no path has to be linear, as probably a lot of the times you think like, oh, if I want to do this, I need to do X, Y, Z. My path was actually pretty unusual. I started studying in undergrad marine biology. That was what I was going to do for the rest of my life. I was really inspired. So I'm from Portugal. 90% of the country is facing the water, and I really, enjoyed the idea of like saving the ocean, I guess. And during that time, I actually was very fortunate to be doing 1 year abroad in Rome, and I did some traveling by the sea and did some prospect research in the by the ocean. But then for my final thesis, that back then Portugal, the undergrad was 5 years, so the last year we had to do a thesis, and I joined the lab of molecular genetics in Portugal, in Porto, and that really changed my approach, I guess, or my understanding of what biology could be, mostly because I really liked the ecology and the environmental part of marine biology, but scientifically, it was really not enough for me to like describe where things are and like where fish reproduce or the behavior of this versus that. Whereas when you get to the lab, you actually get to change things very quickly. You can put GFP in proteins, you can see where they are, you can knock out a gene, you can overexpress, and that really became something that I was very fascinated about. And so from then on, I kind of thought like, even though it may sound a little counterintuitive, I thought, you know, leaving the ocean and actually starting working in the lab was my calling. During that time, I was also working for a year. I worked in environmental education, teaching kids about the environment and the coastal and all that. And then I was very lucky and fortunate to join a PhD program in Portugal that basically allowed me to go anywhere in the world. So we had this scholarship, and you could just send an email to a professor and say, you know, I have my own money. Do you like me? Can I join your lab? And so I did my PhD at UCSF in San Francisco, studying stem cells. And at the time, I also really wanted to focus on stay in developmental biology. That was kind of like my passion. And once I finished that, I also started becoming really interested in how the way the nucleus is organized and everything that really revolves around the nucleus and how that really instructs cells to become what they are. And so I joined the lab of Dr. Emily Bernstein at Mount Sinai to be, you know, at some point meeting David here. And initially we were working a lot with things that are related to what I was doing as a PhD, and I talk a little bit about that in a few slides. And so we were working on IPS reprogramming, looking at histone variants. But at that time, my PI said, you know, why don't you also apply to fellowships, because, you know, nothing comes for free. So there was this opportunity to work on breast cancer. I initially never wanted to work on cancer, and I kind of resisted, but then, you know, it's a grant, sure, well, we can just do that. And I actually got the grant. So all of a sudden I have to work on breast cancer. And that really also changed my career in a bit, because then I was really considering myself like a very basic biologist working on embryonic stem cells, the concept of embryology, and then all of a sudden I was studying breast cancer. And then when I had to start my own lab, I focused a lot on a lot of these questions in terms of cell identity, but really focusing on how cancer cells really use those same pathways of developmental biology to, you know, for their own benefit. And also during the pandemic, because we had resources, my lab actually started becoming really proficient in doing single cell sequencing, and so that also kind of took a turn on my research. So you can see that, you know, no path is really defined, and I hope yours might also take some turns. And my only advice is, like, you know, if you just follow what you are curious about in science, I think you'll be okay. So this is what I usually present, just to kind of give you a broad introduction of like what we study in the lab. This is what I show to graduate students in my first class of genome biology. This is the genome. This is a small portion of the genome. And as you can see, you know, it's pretty complicated to identify particular regions in the genome. And so what the epigenome does, or like chromatin biologists focus on, is in how the genome is annotated, right? So, you know, if you have a pen, you annotate the region, in this case, oops, sorry. You annotate a region in the genome where you know that there's actually a coding gene. And that can be done by histone marks that really basically cover that specific region. But also it's important for the cells and for the nucleus to be organized in such a way where the starting and the ending is annotated as well. So there are specific marks that also annotate the transcription start site. Another very important feature of this kind of annotation, or this code, is to annotate genes that have to be active versus genes that have to be inactive. Because as you probably are aware, with a very few exceptions, all the cells in our body have exactly the same genome, and they don't all express the same genes. And that is basically done through a lot of pathways, but also is locked in the epigenome as a way for the cell to know who the cell is. And so repression and activation of the epigenome is quite important. And then this is the part that specifically my lab is very interested in, is these regulatory elements that could be actually further apart or really close by the gene itself. And these regulatory elements are basically sites where transcription factors bind, and the binding of those transcription factors affects the way the genes get expressed. And so in my lab, we're very interested in trying to identify all these regions in the genome And these are very importantly context dependent. So different cells will have different regulatory elements active, and those regulatory elements will be bound by different transcription factors. And so it really becomes very complicated, all this regulatory process. So this is how we see the epigenome. Of course, you know that the DNA is not going organized in a naked shape or form in the nucleus. It is actually uses these proteins called histones that form the nucleosome that basically wraps the DNA around them. And these histones also have tails that protrude out, and they have specific residues that can be modified. These are the marks that I was alluding to in the beginning that give the green or the red or the blue coloring. So different marks will actually allow the genome to be understood differently. There's also the DNA itself that can be methylated or hydroxymethylated, and this also affects the way the genome is read. Typically, methylation around the promoters means repression, but in the gene body actually means something else. So the beauty and the curse, I would say, of the epigenome is really that it's extremely context dependent. And so it is really important for one to, like when we're studying something, really identify a specific model and stick with it to actually be able to take the conclusions that you are trying to do. So here what I'm basically showing, I don't know if it translates very well, but this is like if you pile the histone marks or those histone modifications here, if you pile them in the genome as an average, you can start seeing, I don't know if you can actually see the nomenclature, but this is H3K27 acetylation that marks enhancers. It also marks promoters. There's H3K4 trimethylation right around the promoter itself. This is the blue color that I was annotating over there. The green color, for example, around the gene are marks such as H3K79 dimethylation and K36 trimethylation. So you can see that there's a lot of different marks that can actually help us annotate the genome. But it makes it much more complicated because marks will represent different things. So a couple of years ago, my lab actually was very interested in identifying different patterns for these regulatory elements, or enhancers as we call them. And we published a paper where we showed that if we basically see these enhancers as marked with different flags, you can actually see that, you know, there's different kinds of enhancers. There's the ones that are really close to the promoter, and actually those probably would just be annotated as promoters. There's active enhancers, so marked with H3K27 acetylation. The ones that are repressed with K27 trimethylation. Some that we call ATAC only because they are open chromatin regions, but they don't have any other marks, but they are still active. And then we identify this class that we call macrobound enhancers because they have this histone variant that seems to be very specific, and it has a very specific regulatory influence in these enhancers. So as you can understand from this graph here, 1% of our genome is encoding for genes. But actually, if you look at the percentage of regulatory elements, it goes almost like around 8% for the human. And if you actually think about the fact that those enhancers are much smaller than genes, it actually, you know, it's a lot of enhancers that we are interested in looking at. And as I mentioned, we can look at these enhancers looking at specific histone variants that mark them. We can look at DNA methylation patterns. We can look at which transcription factors bind to them, and that really affects how they are read, and then non-coding RNAs. So when I started my PhD, I was very interested in this idea that stem cells could actually be deriving all different kinds of cell types in the body. And just as I was doing my thesis, this group in Japan, Dr. Yamanaka's group, identified a way to actually go from skin fibroblasts by overexpressing 4 different genes and turning them into stem cells. This is the IPS reprogramming that, you know, people currently use. And with that idea that only 4 different transcription factors can actually completely change the fate of a cell, really brought us to this, you know, fascinating new field of biology that, you know, basically can allow us to now define the differentiation of every different cell type by adding different transcription factors. When I was a grad student, my idea and my contribution to this was actually showing that this process of reprogramming really had a lot to do with the way the chromatin was organized. and we identified a chromatin remodel, a CHT1, that was very well implicated in leaving the chromatin open. And so that was kind of like our help to understand this reprogramming process. But now that we are looking at cancer, we can actually think about the reprogramming, or this idea of reprogramming, in a very similar way, because cancer is a way of reprogramming a cell towards something that is more resilient, doesn't really require doesn't abide the same rules as normal cells. Typically, they overcome senescence. They don't really get so it requires all these different cell reprogramming effects. That I like this illustration here because it really shows, like, this is kind of like using the epigenomic epigenetic landscape, where the cells have to choose their path, and in the case of cancer, it really gets to be hijacked and used in the favor of the cells. So when I started my lab at Mayo Clinic, I was very interested in mapping these regulatory elements to start really trying to understand how cancer was using these regulatory elements to its benefit. And so we started using this technique called assay for transposase accessible chromatin, or ATAC-seq, that was developed in 2017. And this basically makes use of this transposase called TN5 that's hyperactive. And if you add this transposase to the genome, it really starts incorporating the DNA at random places. But if you give this to a whole nucleus that is structurally defined, the TN5 will only have access to the regions that are devoid of the histones or the nucleosomes. And so when you actually use this TN5 for like 30 minutes, for example, you will start picking all the events that happen just at regions that we call open chromatin. So those are the enhancers and promoters. And because we can actually, this transposase, every time it does incorporate, adds little, it can add little barcodes, we basically make a library out of this TN5 transposition, and this library is basically showing us all the events that happen that are associated with open chromatin regions or enhancers. And so this was a paper that I was showing that we can do this across different tumors. At the time, I got some funding to do this in ovarian cancer, so we started working on that. But then it became also very clear that looking at open chromatin in different tumors can be very interesting, and there's a lot of differences between different patients. But then we really started trying to understand whether we could do the same thing at the single cell level to look at different heterogeneity. As you can probably imagine, and you probably are aware of, when you treat the cancer, whether it's radiotherapy or chemotherapy, depends on the tumors, most of the cells actually die. So the treatments that we are currently using, for the most part, are very effective. The problem comes with the details, right? And there's a few cells that will actually not be killed by the treatment. In the case of ovarian cancer, which I'll explain a little bit later, the path for treatment is always very similar. It's chemotherapy, and when you treat the tumor with chemotherapy, most patients will respond very, very well, and for 6 months they will be disease-free. But then there's maybe these cells that I'm illustrating here as green, or drug-resistant cells that don't die. And so our idea, when we started looking into the idea of understanding tumor heterogeneity, was to try to understand whether epigenome, or the way that these cells are organized, the nucleus is organized, whether we can learn something about them and potentially define things that what I call here resistance-specific enhancers. Are there any regions that are very important for these green cells to actually survive chemotherapy? And if we understand those, we could potentially develop new therapies. But the most important part here that I want to highlight is this concept of transcriptional dependencies. If we understand what are the transcription factors that are specific to these green cells, we could potentially really treat the tumor in a better way. So what we started to do in the lab was using some of these approaches that just started being available the last 5 years, or a little bit 10 years. Single cell sequencing has really kind of exploded as a field, and we were, as I briefly mentioned, during the pandemic, we actually were very fortunate to start using some of these platforms because we wanted to look at that in the context of COVID-19. But in my lab, we also started doing that to understand cancer. So I'm highlighting here a few different platforms that allow you to do not just expression, for example, which is single cell RNA-seq, but also incorporate different modalities. And again, because we were very interested in the way the genome is organized, we wanted to include not just expression, but also some chromatin analysis as well. And single cell ATAC-seq was becoming available, and so this new platform that at the time was coming out really allowed us to isolate the nucleus and look at expression and ATAC-seq from the same cells. There's obviously a lot of interest in developing novel platforms, such as, like CytSeq is probably the first multi-omic modality available, where you actually add antibodies, and so you have cell surface markers and transcription characterization. You have nowadays, obviously, a very interesting field on spatial transcriptomics, so try to understand a lot of these features, but within with a spatial context, which allow, for example, pathologists to really understand the disease in a better way. And then there's features like looking at the epigenome and also looking at mutations that come alongside, and also other modalities. So this is basically the platform that we're using. We're looking at, it's called Multiome, and we isolate nuclei, so we can do single cell ATAC-seq. We look at all these different regions that are open chromatin from different cells, and then we do the same thing for RNA. And this importantly, is done from the same nuclei. This is just kind of like how the platform works. You have these gel beads that have a barcode, a specific barcode that is unique, and then you isolate nuclei, you do the transposition first, and then when you have all the TN5 marking or breaking your DNA, you bring these barcodes together, and you have what we call the gems, or the single nuclei gems, that basically indicate that every single cell has a different barcode. And then once you do that, you can basically separate the cells and develop two different libraries, one for the ATAC-seq and one for the RNA-seq, which is here the expression library. So these are a few projects that we have been developing using these technologies, all or most of them all related to ovarian cancer. But today I'm going to really focus on identifying these green cells, which in this case we're calling them persistent cells because they are the cells that persist chemotherapy. We're also obviously very interested in understanding how the heterogeneity also affects the tumor microenvironment. And we also have a project in transposable elements, but I'm not going to really talk about that. So let's just talk briefly about ovarian cancer. In the US, ovarian cancer is actually not one of the most prevalent malignancies in women, but if you actually see, once it comes to lethality, it actually ranks much, much higher. And the reason for that is that if you actually look at the mortality to incidence ratio, the ovarian cancer is really up high, very close to, for example, pancreatic cancer for global general population. So there is an urgent need to actually understand ovarian cancer because it actually is very lethal. One of the reasons for that is that when we detect ovarian cancer, it's actually in very late stages, and it's very hard for us to have good ways to detect ovarian cancer at early stages. One thing that, if you're not in the field, most people don't appreciate is that actually ovarian cancer does not come from the ovaries per se. It actually is mostly a disease of the fallopian tube. So the fallopian tube cells will actually start transformation and will eventually migrate to the ovaries. And this, there's a lot of groups that are very interested in understanding this tropism because somehow these cells are really attracted to the environment around the ovaries. But the disease itself actually comes from the epithelial cells from the fallopian tube. And it has a very striking p53 signature, especially high-grade serous carcinoma, which is what I'm going to be discussing today. So most high-grade serous carcinoma, which is the most lethal, have p53 mutations. And then there's these things that we call stick lesions that are basically pre-malignant stages that are found in fallopian tube prior to the actual full-blown disease. I'm not going to really get into that many details molecularly about ovarian cancer, but this is kind of like just an overview of the molecular landscape of ovarian cancer. There's basically two different types of high-grade serous carcinoma. One we call homologous recombinant deficient. These have BRCA1 mutations or BRCA2 mutations or repression of BRCA genes. BRCA is a well-known gene also in breast cancer. And then the ones that did not have these deficiencies. So basically we can divide ovarian cancer or high-grade serous into homologous recombinant deficient or proficient. So when we started doing this analysis, we got in, a big collaborative project, including Raj Khanna, who was basically developing a biobank to isolate fallopian tube from non-malignant patients. So, for prophylactic surgeries, some patients would remove their fallopian tubes, and we collaborated also with Dr. John Baroja, both at Mayo Clinic, to really get our hands into the high-grade serous carcinoma. What we basically doing here is, as soon as the surgery happens, we get the samples, we freeze them in our special protocol, but then we are basically able to isolate these cells, isolate the nuclei, and get the samples. And so for high-grade serous, for example, after the bulking surgery, my lab is on call to collect the tissue itself. Well, when we collected the tissue, we started collecting both patients that were chemo naive. We call them chemo naive. They never seen any chemotherapy. And also there's a few patients, and now depending on the protocol for different hospitals, some patients actually start getting treated with chemotherapy prior to the debulking surgery, just to shrink the tumor before the surgery happens. And so these we will call neoadjuvant or NACT. We'll see NACT quite a few times. And so the neoadjuvant patients will basically represent what we call the residual disease, so the cells that survive chemotherapy in the first place. And this was work done by my computational biologist, Wasim, an MD PhD student, Mihai, and another postdoc that left the lab that started this project. So this is kind of like the cohort that we use. We use 5 different patients that are non-malignant. We call FT for fallopian tube. Then we have 6 patients that are naive, so never seen chemotherapy, and then 3 that actually were already resulted after chemotherapy. One important feature is that these are not paired, so we don't have any samples that were before and after chemotherapy. So I don't know if you're very familiar with how single-cell analysis is done, but basically you can use these UMAP plots to basically plot every single cell. So a dot here is one cell, and then you can plot them all together to show the diversity of the different cell types. Because they have different gene expression patterns, we can actually annotate using specific markers. We can annotate the kind of cells that they are coming from. One important thing that you can see here, so here I'm using basically expression, JEX, or ATAC-C. And these basically are very similar in pattern, but then you can actually incorporate both the open chromatin and the transcription in what we call the weighted near neighbor plot, which is the JEX plus ATAC-C here. And you can see that there's a lot of different cell types: T cells, NK cells, B cells, macrophages. And then in pink here are the epithelial cells. These are the tumor cells if we're talking about high-grade serous, or the fallopian tube cells if we're talking about the non-malignant. And you can see how these cells are distributed by class and also by patient. One important thing that you can actually see when you look at here at the patient distribution is that these different clusters are all coming from different colors. Those are all epithelial cells, and that is because when we're looking at single cell, there's a very strong patient specificity. So every patient will have a tumor that it's very unique. When you look at other cell types, for example, here, this area here, which is full of T cells, all the T cells are bundled together because T cells, even regardless different tumor, the different patients, all the cells are going to be much more similar to each other. I'm not going to really need to go into much detail on this, but basically, as I was explaining, we use different expression markers to annotate the different cell types, and this is kind of like how you can look at different patient samples and the contribution of different cells from different patients. What's really strikingly seen here is that the majority of our high-grade serous are all pink, so that's epithelial cells, and you can see that in the normal case, there's actually a lot of different cell types. One important thing that you need to be aware is that high-grade serous, for example, has been really a very poorly, a tumor that's very poorly used in immunotherapy. So immunotherapies don't really work in ovarian cancer, and that's something that people are really interested in trying to understand. And one of the reasons for that is that we call these ovarian cancer tumors cold tumors, because they don't really get so much incorporation of the immune system in them. When I started this project, obviously my idea was that if you incorporate different features such as expression and open chromatin, you will be able to identify those green cells that I was showing in the beginning very easily, right? Because now you're adding different modalities, these cells are going to be very easy to identify. And that was not the case. So when you now basically take all the epithelial cells that we had been collecting, we remove all the cells that are not epithelial cells, so both the cancer and the non-malignant cells are here. You can see that there's different clusters, but none of them was actually associated with, for example, resistance or survival. One thing that we actually noticed very nicely was that there's this group of cells that we call cycling cells, and those are highly relevant in naive patient samples, but when you treat them with the neoadjuvant chemotherapy, they're almost gone, which makes sense because these therapies, chemotherapy specifically, targets cycling cells very well. The other important part of this initial analysis is that we can start looking, because we have both ATAC-seq and RNA-seq. You can look, for example, of markers, in this case, a marker for ciliated cells. Ciliated cells are part of the fallopian tube, and there's ciliated cells and secretory cells, those two different cell types. And you can actually look and infer different regulatory activities. This line here is basically what we call a connection between an enhancer and a gene, and these can actually be done because you're accumulating those open chromatin regions and associating them with genes that are expressed in that specific cell. If you put them all together and you compare the probability of a ciliated cell to have an open chromatin region here and this gene being expressed, you create a link. And so these links start becoming really useful for us to understand what is regulating a specific gene. So initially we were comparing non-malignant, so fallopian tube samples, with high-grade serous naive, so cells, patients that never seen chemotherapy. We saw a lot of different things actually happening, obviously, because with the tumor, we can start understanding exactly what are the pathways that are very highly involved in the process of malignancy. And some of them make a lot of sense. So for example, MYC targets, things that are associated with cell cycle are very high with tumor. Another feature that people have shown in ovarian cancer, and we can validate here with our approach, is to show that there's much more macrophage, so monocyte differentiation in the context of ovarian cancer in comparison to the fallopian tube. But because we know, and there's actually already been so many studies in single-cell RNA-seq in ovarian cancer, we really wanted to incorporate these data sets and try to understand a little bit better what happens to a lot of the transcription factors and how they really help each other to develop a tumor. So we compared, for example, so when we're looking at here, we have all these ATAC-seq peaks, and for each transcription factor that we know binds to a specific region, we basically give it a score. So if for example, a transcription factor is binding to all these 3 peaks, would get a higher score than if it only binds to one of these peaks. And obviously, this is just a representation. We're talking about tens of thousands of peaks. And so the score, you can score basically transcription factors per cell, and then you can actually compare the signal or the enrichment from transcription factor to transcription factor within a specific group of patients. So fallopian tube, you can actually see how correlated transcription factors could be in this kind of context, so in the non-malignant, and then in the high-grade serous naive context. We don't really need to get into much detail here, but there's some transcription factors that we know are involved in ovarian cancer, such as MECOM, WT1, PAX8, and SOX17. They are very important in fallopian tube, but then once they get in the context of malignancy, they actually kind of separate into 2 groups. There's MECOM, PAX8, and WT1, together with actually a stem cell marker SAL4 in this cluster, and SOX17 starts using different partners to actually do its business. This is kind of like that kind of approach where we're now able to compare transcription factor activity with the resulting or the subsequent regulatory effects in different genes. One thing that you can actually do from this kind of analysis is trying to define whether transcription factors are acting as repressors or activators, because the binding of the transcription factor toward in a regulatory element doesn't mean that actually is going to be activating. There's a lot of transcription factors that are known to be mostly repressors. And we actually, with this kind of data analysis, we see that depending on the context, again, the same transcription factor can actually be activating and regulating, and the reason for that is because it's actually having different partners, and that's makes it very complicated. But so one interesting feature that we collected from this analysis is that you can see that the majority of the big nodes of transcription in here are activating. But then when you actually look at the tumor, you see that a lot of transcription factors actually now are being repressing. So there's definitely a shift in terms of like how transcription factors are regulating different pathways. Here we basically lined these plots together for each transcription factor that are aligned here, and you can see that the same transcription factor in the context of fallopian tube has mostly green in this case, but then in the context of cancer it becomes red. So an activating transcription factor becomes a repressive transcription factor. So then what we actually try to understand is, can we understand a little bit of the biology of these residual cells, the cells that survived neoadjuvant chemotherapy, and compare those to what we call the naive context. So comparing these cells that never seen chemotherapy with the residual cells. So in this comparison, we first look at expression. So we look at genes that are differentially expressed, downregulated on the left, upregulated on the right. And you can see that there's, obviously a lot of different things happening. There's a lot of genes that change. And here we're comparing the 6 naive versus the 3 neoadjuvant. But then when we actually looked at different papers that had been shown before that, with signatures that they would identify as, good for prognostic or, associated with specific cells, a specific subtype of ovarian cancer, you can see that the results are all over the place. So the neoadjuvant, like the signature that is upregulating the residual disease, could really define anything in expression at the expression level. What was really interesting to us is that when we look at the ATAC-seq portion, so open chromatin features, you can start seeing that there's like a very strong enrichment of a lot of transcription factors on the top, so associated with residual disease. And some of them actually were known to us, such as PAX8, SOX17, MECOM, WT1, which I mentioned before. So these transcription factors that we know are important in the context of malignancy seem to be also very relevant in the residual disease. So they were playing some important role in the fact that the cells are surviving. And so again, we were trying to understand if this residual disease or this persistent signature was actually something that could tell us, could distinguish the samples or the patients that either were going to become sensitive or resistant, even though they never seen chemotherapy before. So these 6 patients that we collected that had never seen chemotherapy, 3 of them actually went on to become sensitive, so they responded very well to chemotherapy, and 3 of them were actually poorly responding. So after 6 months, actually the disease already was coming back. One important part, and this is more for people that study ovarian cancer, is that the debulking status was optimal. One of the main variables in response to the chemotherapy is the fact that you actually are able to remove most of the tumor, and all of those were at the same level. So we were excluding this variable in terms of like resistance and sensitivity. So our question here was, if we compare our residual genes, the genes that are important for expressed in the residual disease, and compare them with the ones that are resistant to chemotherapy, even though they're naive, there's a few genes that are on that list, but the overlap is not that striking, as you can see here. Whereas when we actually look at the ATAC-seq portion, the transcription factors that are active in neoadjuvant are actually very similar to the transcription factors that are active in resistant. So even though these cells never seen chemotherapy, the same transcription factors are going to be important or have some kind of role in making these tumors resistant. And so this signature that we call the epigenetic persistent signature was revealing to us by looking at the heterogeneity with single cell analysis. So with this kind of idea in mind, we basically developed a pipeline to reduce our number of transcription factors. As you can see here, we were close to 600 transcription factors in terms of activity that are very similar between neoadjuvant and resistant. So we performed this sorting some of the activity of the transcription factors between the neoadjuvant and the naive, and then we made sure that at least 1% of the cells were expressing these genes to actually come up with this, what we call the final persistent epigenetic signature, which includes 178 transcription factor activities. We can then use the same approach of like mapping all these 178 to the ataxic peaks, give them a score, and basically give a score of this persistent signature to every single cell. This is a general list of all the different factors that we have identified that we believe are part of this signature. There's obviously transcription factors, most of them are transcription factors, and then there's a lot of them that are chromatin regulators, so things that still bind to DNA but they don't recognize DNA binding. And so what was actually really pleasing for us is like now that we are able to give a score, a persistent score to every cell using this epigenomic signature, you can actually see that if you look at the sensitive cells, so we're basically putting all the patients together that are sensitive, all the 3 patients, and you see that the score is the lowest here. The resistance has a middle score, and obviously the neoadjuvant has the highest score. But they are all significant. So between the sensitive and resistant, there is definitely already a propensity for these cells to be resistant to chemotherapy just by looking at their epigenome. So people have been studying this persistent signature or trying to understand what specific features are involved in this signature. There's a study from 3 years ago that showed that by looking at just single-cell RNA-seq that there's these stress-associated genes, so a lot of transcription factors and other genes that are involved in stress response are the ones that seem to be very important in this survival of these cells. And they actually show that if you look at the expression of these genes, there's a significant p-value, so the signature of stress response is able to separate the response. In this case, they actually had a very nice cohort where they had primary tumor and then debulking surgery, so they had pre and post, so it's a longitudinal study. So we were interested in this stress-associated signature that they had found, and we basically go back to our the ability of mapping these links between enhancers and gene, and for each gene we can actually annotate whether we have an activating connection or a repressing connection per transcription factor. And so if we're showing here green, means that the majority of the connections for the specific gene are activating, and if it's red, means that the majority of the signature that we identified connects with repression of this specific gene, and then even is yellow, and if it's gray, basically our signature does not identify any links associated with that gene. And so what was interesting is that, if we look at some of the features that people have shown before, stress-associated comes on the top as, the most regulated, one of the most regulated signatures of our signature, of our 178 genes, and then you can actually look at specific genes that are part of this stress-associated signature, and you can see that the majority of these stress-associated genes are in green, so they are activated by our signature. And the same thing when you compare naive and non-naive. And so this, for me, was basically my favorite illustration, or the time when we were considering like putting all this data together and thinking about like, is this really real? Like we have these associations of 178 transcription factors. How do we know that they actually are all doing something important for the cell, right? Because there's a lot of transcription factors. Usually people identify, a couple of transcription factors or a couple of genes that are really important. In this case, we are talking about potentially an orchestration that it comes from a lot of different players. And so when we looked at the naive context, you see all these lines here are part of our 178 signature. And they are kind of like all over the place, and if you see the correlation between different genes, that there's a wide range. Once you look at the neoadjuvant context, you see that they actually, most of our epigenetic signature is clustered into these two different clusters. So there are clusters of transcription factors that seem to be really doing the job that it's required for these cells to survive. And then we wanted to know, because we didn't have longitudinal samples, we wanted to know what happens before and after chemotherapy treatment. So we turned to a PDX model system. Patient-derived xenografts are basically these immunodeficient mice where you can actually put ovarian cancer, ovarian tumors from patients, and they grow these tumors as you know, in vivo, and we can actually treat them with a very similar dosage and approach that we do for ovarian cancer. So carboplatin and Taxol are the standard of care for chemotherapy, and we've got 3 different tumor types. One that we call the sensitive, because after treatment, 38% of the tumor, you only get 30% of the tumor, as represented here. We have one that we call stable, because the tumor didn't grow, but it also didn't regress. And one that we call resistant, because basically, even after chemotherapy, the tumor continues to grow. So we basically had a little bit of all situations. One important thing that we wanted to do is to understand whether the cells that actually are resisting to chemotherapy are specific clones. We can do that by looking at mitochondrial DNA, because mitochondrial DNA, even though it's mostly removed from our analysis, it's very small, so we can actually do genotyping using the mitochondrial DNA to look at specific variants. And if those variants are always present amongst all the cells that we have, we basically identify that as one clone. So in this analysis, we basically did not find any specific clones that came that were arising from this treatment. So in a way, this basically is just telling us that whatever happened to the cells was epigenetic and not defined by a genetic mechanism. And that people have shown that before, that a lot of the times the resistance to chemotherapy it's not identified by specific mutations, but it has other sources. So we did the same thing. We calculated the score for our different single cells, and as you can see, the similar plot shows up here. So we have the sensitive with the lowest score, and then the resistance with the highest score. And this was really obviously very rewarding for us, because it basically validates that even in a longitudinal test, we can show that these the tumors, even before they saw chemotherapy, the resistant have the highest score. I don't know how we are in time. So yeah, so I'm just going to wrap up. Basically, with this single cell epigenomic approach, we were, as I pointed out initially, we were trying to identify these cells that are persistent or resistant to drug treatment. We performed this analysis to really try to understand how we can learn something from the residual disease and try to implement that and potentially understand epigenetic signatures that can tell us whether the cells are going to be resistant to chemotherapy. And then, very importantly, we really are starting to narrow down to this kind of rewiring of the expression program that is driven by these transcription factors. And this is, the transcriptional dependencies that I was talking about. And so, I mean, I'm pretty sure that you guys all have to deal with these computers trying to prove that we are humans. And basically what we're trying to do here is using a similar approach. Like, can we identify cells from a pool of cells, completely random? Can we identify the cells that are going to be surviving chemotherapy? We're not quite there yet, but we at least, if we put them all together, these green cells, we believe that we are finding some signatures that are going to be important. Finally, I just want to thank my lab. This is a view from the downtown campus. This is the Mayo Clinic. We have a bunch of collaborators that obviously are very important, and I mentioned them before, Raj Kannan and John Roja. This is the lab. As you can see, people all over the world come to join us, and this is the funding for the lab. And then I wanted to give an embarrassing photo of David when he was trying to find samples in the liquid nitrogen. This is David maybe around 2015, 14. Yeah, because you can see he was actually that time working in the lab. And I'll take any questions. Thank you.
Very nice talk. Thank you. I was wondering, have you integrated your single cell data with spatial expression in the actual tumors, I mean like in vivo? And if so, have you seen a pattern, like a gradient of maybe cell differentiation pathways? Because I noticed that there are several transcription factors that are very important for these gradients that form during development. So I was just wondering if you have that
Yeah.
type of data.
Yeah, so I mean, definitely looking at this in spatial will be something that we will start doing soon. I mean, these obviously are very expensive platforms, so we're moving to them relatively slowly, but we're very interested in that, especially well, the question that you're asking is more like basic science, and it's actually very interesting, and that's probably what would be more appealing for me to actually start studying. But when we are writing grants, actually our biggest goal is to try to understand the microenvironment and using the spatial technologies to see, for example, if our green cells, our persistent cells, are more close to a niche that it's immunorepressed. But those pathways will be very interesting to also understand, because then you can start seeing which pathway really is required for this resistance or stress response association. Yeah, we haven't started with the spatial yet.
Thanks.
Two questions from the deep ignorance. One is, one expects a lot of heterogeneity in tumors. So you're working with 3 patients or 6 patients. Is this enough? I mean, do you think that this, once it gets huge, with a lot of data from other labs, and it's going to be similar, or it's going to change a lot?
Right. I mean, the short answer is definitely not a lot. Definitely not enough. I mean, this was, I guess I can say, call it a pilot project, because we really wanted to start understanding the features. From what we gather in the literature, I mean, most single-cell RNA-seq patient cohorts go around 20, the maximum. So I mean, it's still we're still really growing. And one of the things that, as I alluded in the beginning, there's 2 major molecular subtypes of ovarian cancer, of high-grade serous, HR deficient or proficient. We actually have no idea yet, and we're actually working on that, try to understand whether we somehow picked a specific one. I mean, it's 50-50 chances, so I'm pretty sure that some of them will be. So it's definitely not enough. What is interesting, at least in the concept, is that we can see that by expression itself, you can't really explain the whole thing, but there is some kind of propensity that it's epigenetic that will allow us. And I mean, it all comes down to, once we have this, we can validate it with more samples. One thing that we are actually currently doing, as we actually just submitted the paper, is to compare that to, for example, other signatures from there's a paper that actually only did single-cell ataxia. And we actually see the same thing, that now that we know where to look for, we see it as similar. And that's a good validation, because, this is a different platform. It's only in ATAC-seq, of course, so we don't know exactly the same, we don't have the same resolution, but it shows us that, the signature is still there. I'm still very skeptical about the fact that we still have 178. And, I've been telling my postdoc, can we cut down this to like 50 or something? And I mean, the reality is, I mean, we actually even did a rendition approaches, and maybe with more samples we will be able to narrow it down. Or we actually will identify patients that are going to be, favoring those 60 versus the ones that will favor those 70. So yeah, it's a work in progress.
Yeah, this is a little bit the other question that is also from ignorance, of course, that because we have a lot of students here from the pharmacobiology department also interested in the use of the discoveries, the knowledge, the practical use of the knowledge. How do you work with 65 transcription factors? I mean, how do you translate that to something that can be targeted, exactly, because it's like a huge net of things, no?
Yeah, so it's, you don't, right? Like at this point, this concept, and I mean, I'm at Mayo Clinic, so every time I reach out to clinicians that treat patients, they're like, oh, this is really cool, but I have no idea what I'm going to do with this analysis or signature. And obviously at this point, all we can say is that, potentially there's a benefit to understanding whether some patient, if we were able, for example, to do this sort of sequencing for every single patient that enters the clinic, whether this patient has an epigenetic propensity to be more resistant than not. If that's the case, then probably we could put them in a different kind of drug regimen, right? That's at this point is really the only practical thing that one can get from this. But I do believe that once we define the signature better, we will potentially find things that will tell us, well, if it's going to come down to 10 prescription factors, we should target those. So one of one of the genes that we are actually very interested in studying is SOX2, which was part actually of my PhD because I was doing reprogramming, and it's a reprogramming factor, very important in stem cells. It's actually highly relevant in this context, even though it's very lowly expressed. And that's probably one of the reasons most people haven't identified it. And so, you know, SOX2, for example, could be one of those.
Just the last thing, these persistent cells, would you call them cancer stem cells?
So initially we were actually going after cancer stem cells. And the concept could be very similar. A lot of the genes that are in the persistent signature are stem cell genes. And so one could call them cancer stem cells if one wants. At this point, we wanted to stay away from that classification just because, at this point, all we know is that these cells exist in the tumor and they have the propensity to persist after treatment. But clearly the cancer stem cell signature is there. We see SOX2, SEL4. A lot of the genes that are part of this signature are stem cell genes.
Thank you for your talk. Have you ever looked for any specific cell surface marker that is actually expressed in those persistent cells so that you can use them to isolate and validate that these cells are responsible to develop the tumor?
Right. So that's an amazing question that we are trying to deal with. So for example, coming back to the idea of cancer stem cells, so there's cancer stem cell surface markers that we could actually try to see if they match this signature. The biggest problem is that when you look at by single cell expression, those expression markers like LDH are poorly expressed. And so we can't really say that the persister cells are high on that. If we had probably a third modality where we actually look at protein or using antibodies, we could potentially get to those questions. So try to understand whether by protein or cell surface markers we could distinguish persisters and not. At this point, with what we have, we don't really. I'm not comfortable saying that we actually were able to identify. But that's really, at the end of the day, it's also very much more translational. People will want to know what these cells are, not just like a very expensive modality of RNA plus say tac-seq that will tell you, because that's not going to be useful for anybody, except for me to write grants or to graduate students, exactly, which is very important.
So I was wondering if this mechanism of resistance, which I would call intrinsic resistance, somehow applies to other type of drug resistance, let's say epilepsy or other types of cancer. And this mechanism, or this is very specific for ovarian cancer.
Right. So the short answer is I don't know if it's very specific. As I alluded, everything is context dependent in the epigenome. So it's possible that some of these features will be applicable to other tumors. One of our projects is actually try to see if we can still see the same thing, for example, in breast cancer. One thing that actually I did not put here is, even though the signature that we have in the PDX models before treatment shows a nice correlation between resistance and sensitivity, we actually see in the tumors that are sensitive, we actually see an induction of the same signature. So even though the resistant tumors intrinsically have that signature, we also see an increase of the signature in the ones that are sensitive. So there is both. There is a little bit of inherent capability of resisting, but there's also an induction by, for example, activation of stress-associated genes. So we actually see a little bit of both. It's not just intrinsic. Cells do tend to try to fight the chemotherapy in a way that we can see or read in the epigenome. So it's a little bit of both. It's possible that some of the cells already have that capability open in the open chromatin, and so they are able to resist the treatment. Others will try to use the same pathways or the same signature, but they're probably too late, and so by the time they get the treatment, they will all die. So we see a little bit of both.
So in the second group, where they are sensitive to treatment, but you see an increase in the signature, is it related to the drug or to other factors? Because you mentioned the stress.
Yeah.
How about the drug?
The short answer, I have no idea. For example, for ovarian cancer, we could try something similar with PARP inhibitors, because they are also used in the clinic, and see if PARP inhibitors also give us that same signature. But then, we would have to try different tumors radiotherapy, for example, for, there's different pathways, and obviously this persistent signature might not be the same for different drugs because, the cells will respond differently. But that's a very good point. Thank you.
Any question? Oh, I have a couple of questions. One more sort of basic. Like every time I analyze like ataxic, chromatin marks, whatever, and compare it to RNA-seq, you don't see like a match as you would expect, by the classes that we give teaching students that chromatin marks are related to transcription. I always thought like, well, this is bulk RNA, bulk ataxic, maybe like, RNA stability, whatnot. But in your case, you have RNA from the nucleus, from the same cell, and still it seems that you don't have, this expected correlation. So what gives?
Yeah, so by the way, David was the one that got me started into bioinformatics. So because when we started in the lab, when I was a postdoc, I had no idea, and he was the one that really helped us. So thank you for that. It's actually you're completely right. I mean, it's not very direct to associate open chromatin or activity to expression of genes. There's definitely a lot of confounding factors for that. Like for example, there are some genes that are very small and some genes that are very long, and that when we analyze RNA-seq and open chromatin, that actually really affects the way the signal of open chromatin can actually be measured, right? Because what we do at single cell level is calculating a gene activity by ataxic peak. So we kind of try to identify how much the chromatin is open in a specific gene. And if you do that, not all the genes that are highly expressed have a lot of open chromatin. That's what you were alluding to. The biggest problem with single cell, even though it's from the same cells, is that the data for ataxic is very sparse. I mean, we have, for a particular cell, we probably have 3,000 cut sites, we call them, or open chromatin regions. And clearly, in the genome, if you do this for bulk ataxic, you will get 100,000 or way more. So the only trick we can use is to basically put them all together and use this pseudo bulk analysis. And so we get into the same problem that you guys get when you do bulk analysis, is that the correlation is not very good. The way to circumvent that, for example, for open chromatin, is instead of actually looking at gene activity by open chromatin, you actually are looking mostly at events that are associated with transcription factor binding. So, for example, if you have open chromatin, 100 open chromatin regions, and you know that 50 of them are associated with transcription factor, now you actually start having numbers to analyze, and that's how we look at this persistent signature. The signature is actually not the expression of these transcription factors, is how many open chromatin regions are associated with the binding, potential binding of that. So there's a lot of tricks to try to get some some kind of signature to it, but it's still a puzzling thing that we can't really identify expression and open chromatin very tightly correlated.
And the second one, have you tried machine learning algorithms to try to sort this out? And I'm thinking probably random forest or something that can actually pinpoint to specific factors. Maybe you can that way get to 50 or
right. Yeah. So I mean, we tried. I mean, with my computation with my postdoc, we actually started considering some of those. But I mean, again, the data is very sparse, and with 3 patients from one and then other, it's actually you don't have enough number of cells to actually have a robust machine learning approach to identify these. But with the more numbers, we'll potentially have that capability.
If there's no other question, thank you a lot.
Thank
it was
you.
a very interesting topic.